like SARS-CoV-2 have been detected in wastewater, influenza, and other respiratory viruses. Also, other viruses like hepatitis C, polio viruses have previously been detected from the wastewater. And so when we talk of wastewater data, we are talking of a very complete data that will give you an outlook of infection prevalence and the scale of outbreak in a geographical area. So we know that it's not possible to actually test all the individuals within a given area, but that information can be detected if one focuses on the wastewater. So this data that can be generated from a wastewater surveillance can be used to independently evaluate risk.
Recording in progress.
So how do we do wastewater surveillance, particularly for pathogens? So the first thing which is very important is for you to begin by mapping of the sewerage network. So I will give an example here of the city of Nairobi, Kenya, where we had done a study on SARS-CoV-2, and we started by mapping out the sewerage network of the entire city. And I don't know whether it's can be visible from your side, but those green networks represent the sewerage network of the city of Nairobi. And so after you have mapped the sewerage network, then the next step is now deciding on where you are going to sample. So the sample points, and the sampling points that you are going to decide on should be strategic points that cover specific epidemiological zones or the catchment populations. And so you choose the convergence points where you know that the sewer line will cover a given set of populations. So for example, in what I'm showing here, we had identified about 9 sampling points within the city's sewerage network, including the 10th one here on the far right here, which is a main sewage treatment plant. And from a sewage treatment plant, you would want to sample the influent sample just before it is treated. So I would just take you briefly on how we conduct or the processes that are involved in processing sewage data, sewage sample, and also generating that data. So once one has identified a population that you are targeting, you will realize that within the population, you will have several individuals with various forms of infections. And it's important to note that amongst those individuals, there will be populations that or people that have reported cases of infection, and also there are also those that have not reported cases due to several other reasons. And so the importance of using sewage water is that in the sewage water of a target population, you will be able to cover samples that represent reported and unreported cases. And I usually say, and I make a joke of this, that when you're looking at the sewage, you are actually talking of a sample that is very representative. And I usually say that sewage water is the polling station where we usually get 100% voter turnout. So a sewage water is very representative and covers the entire population. So once the sampling points have been identified, then one can use whatever sampling protocol that you've agreed on based on the study or based on what you are following. And the samples are processed and taken to the labs where the processing involves pathogen recovery and concentration, and then followed with the quantification of pathogens and also even characterization of what pathogens that you find in that. So when you are analyzing sewage data, there are several things that needs to be taken into consideration. We need to look at the shedding rate. We have to normalize the population with the quantities of the pathogens that you generate. And there are also aspects of ethical considerations involved in this. But essentially, that data that you have quantified and characterized can be used to generate epidemiological models that then can be used or reported to the public health officials for action. So briefly, that is the workflow that is needed for sewage water surveillance processing. So I will give a brief presentation on the work that we did with COVID-19 wastewater in the city of Nairobi. So as I've indicated there, we identified about 10 sampling points. And during our sampling, we used two methods. We have the grab sampling and also what I call the auto sampling. So without going into great details, we were sampling over a period of time. So it was a longitudinal study, and samples were processed in our labs at ILRI, and we did quantification of SARS-CoV-2 virus in the samples. And when we profile that and plot out the detection rates, we could see variations of SARS-CoV-2 detection rate over a period of time. But we also did this in parallel with the hospital samples. And when we overlay the two data sets, what we can see quite clearly here is that there's an overlap between the rates that are detected from the sewage water and also the hospital positivity rate. But what is critical in this data here is that if you look, say, for example, when we started to see an increase in the detection rate for SARS-CoV-2, this is on the July 31st. If you look at the corresponding time from the hospital sample, or from the hospital plots here, on the 31st, there was still no significant signal coming out from the population's data. But we had started to see the increase from the sewage data here. So what makes this now so critical is that, as you can see, following a few days later, then the hospital signals began to rise as well. So it is critical to note that one of the utility that we say is we can use the wastewater surveillance for is an early warning system. Because a few days of picking the signal, if this information is relayed to the public health in good time, they are able to respond by imposing several measures that could actually prevent the wider spread of pathogens. So in this context, we say that wastewater surveillance is actually a good early warning system for tracking these outbreaks, etc. So I want to quickly move into the utility of wastewater surveillance beyond COVID-19. As I'd said earlier on, the wastewater is very rich in terms of different pathogens and microbiome at large. And so it is a sample that can be used to analyze quite a lot of pathogens, not just SARS-CoV-2. So, and so this is what I call the wastewater genomic surveillance. And so the early stages of processing the wastewater for genomic surveillance is at the upstream of it is quite similar. It's just a solid particles removal and the microbial concentration. And then the total nucleic acid extraction is performed on it. And so beyond the nucleic acid extraction, that is where now we start to process the samples differently for what I call the metagenomic analysis. So here we can use those extracted nucleic acids to prepare libraries which we can use to do whole genome analysis, or the so-called shotgun metagenomics. And then we can sequence those samples on one of these next generation sequencing machines or technologies, for example, Illumina or Oxford. So for Illumina, as you all know, there are several platforms, but because of the complexity of wastewater in terms of the pathogens, I would prefer using NextSeq machines and above. So once you have analyzed the data, the sequence data that you've obtained from that, then you will be able to detect pathogens that they are in, but also the sequence data can be actually used to quantify the antimicrobial resistant genes within those samples. And this makes wastewater one, useful for pathogen detection and profiling, and also going beyond just detecting the pathogens, but also analyzing the antimicrobial resistant that could be present in a population. So here is a brief data that we have generated so far from the samples that we originally collected for SARS-CoV-2, but we have now extended our analysis to include the entire microbiome within those samples. So as you can see, you get quite a lot of microbiome in it, bacteria and viruses. And when you zoom into these data sets, you can actually pick even some of the pathogens of interest. For example, we were able to pick the beta coronaviruses and particularly the SARS-CoV-2 from that aspect of a metagenomic analysis of wastewater. So this is just, an example to show that, the wastewater can be quite rich, even in picking some minor pathogens within it. But when we focus on now looking at the bigger picture and expanding this to other pathogens and the entire microbiome, I can show you here a presentation of some of the analysis that we are beginning to do, where we are profiling the microbial abundance of the wastewater sample. So for example, here on my left here is a tax abundance of bacteria that we detect at, this is at a family level, and you can see quite a lot. And this is sampling that was done at a particular population catchment over a period of time. And you can see the fluctuation in how the abundance fluctuate with time. Similarly, on the virus here is a tax abundance of virus, again at the family level, and you can see all sorts of viruses that one picks from wastewater. So this is a very rich data from which one can pick or decide to follow some pathogens of clinical importance and follow them through how they fluctuate on the abundances and many other things within a period of time, and use that data to model the epidemiology and present that data to the public health for any action. So I've mentioned of the pathogens, but I've also talked of the antimicrobial resistance. So here again, I'm showing part of our data that we have analyzed from the wastewater following the metagenomics approach. And you can see from the different samples we've analyzed, we are getting several antimicrobial resistant genes. And as you can see from the left, you can see that the sample on the left here has quite a lot of antimicrobial resistant genes, while others from different locations may not have as many. So again, it gives the picture of how much antimicrobial resistant genes are in circulation within a population, and the different populations will show different profiles. So as I get towards the end, I just want to mention some key advantages of wastewater epidemiology data sets. So first of all, I've said that it's a very comprehensive data because as you know, it is not possible to count every case of infection within a population because, of several reasons. Others may not be able to present themselves for testing, etc., or report cases. Others could be asymptomatic cases, and you may not know all this. Others could be having home tests, and may not be reported for consideration. But when one focuses on the wastewater, then you are sure of a very comprehensive data. Also, wastewater, another advantage is that it's very inclusive and anonymous. As I've said, everyone contributes to the sewer, and the anonymous aspect of it is that it removes those aspects of stigma when you are actually testing individuals. The other important advantage is that you get spatiotemporal information. So this reveals the epidata across space and time, because when you pick samples from specific locations, you know the specific area and the population you're covering. And the aspect of taking the samples within a period of time, you will be able to get that data that cuts across space and time. We can also say that the wastewater sample is low cost, is simple, it's fast, and it's less invasive. For example, if you compare to the swabs and blood, etc. So you can take as frequently as possible from the wastewater. And then the other significant advantage is this of this early warning system. So wastewater is a potential leading indicator of new or emerging outbreaks. And indeed, wastewater can precede clinical data reported by several days. In our case there, I think we were talking of about 4 to 6 days ahead of the data coming from the hospitals. So wastewater data, in my view, may be the best way to measure pathogen activity in real time. And then, of course, with advantages, there are also limitations. And so here I am giving a few technical limitations that we know are associated with wastewater when you compare it to conventional surveillance. So one, there is a high level of uncertainty that comes as a result of variabilities. The variabilities are seen in the rate of excretions, pattern of use, fluctuation in the contributing population, and also other variabilities that are associated with the environmental factors. There is also aspects of degradation of the signal in the sewer network. So these are also some of the technical challenges. And finally, we know that the wastewater is a very complex sample. So there are analytical challenges that are associated with the complexity of the wastewater sample. So when I look at the Africa, and I can think of a few challenges that one would face when we focus on wastewater epidemiology. First of all, as I indicated, one needs a very good sewerage network infrastructure. I know that some major African cities would have this, but it's still not spread enough to cover the wider population. So this would be a challenge. And then because of the nature of the samples of the wastewater and the analysis, there is a significant infrastructure, genomic infrastructure involved. And I know that through Africa CDC, a lot of effort is going to improve the genomic infrastructure in Africa. So this, I would say, is still temporary, is a temporary challenge, which may be overcome in the future. And the other temporary challenge is on the genomic analysis capacity. And I know that there's a training that is now going on in Africa to improve the genomic capacity and expertise to be able to use this technology for the benefit of the Africa as a continent. The other challenge I also see here is the ability of the public health to transform themselves to be able to embrace data from this kind of technologies. So transforming public health to be more data driven, I think is still a challenge that needs to be undertaken. So in conclusion, I pick a few things here that I would say would be a good catalyst for effective wastewater genomic surveillance in Africa. I've talked of the need of capacity development, but there's also a need to have standardization and expertise in how to handle, how to analyze the data that comes out of wastewater surveillance. So this needs to be standardized. At the moment, this is not just an issue in Africa, but it's a global issue. Everybody is still developing tools for use, and at some point there will be need to actually standardize these tools and analysis. Again, there is a great deal of strategic coordination that is needed for this to be actually effective. But also, the last point here is the repository function. You will realize that, you know, wastewater surveillance accumulate quite a lot of samples. So if you imagine that you are sampling at almost several sampling points and you're taking samples 3 or 4 times a week over a period of time, say a year, you will quite accumulate a significant amount of samples. And with a significant amount of samples, a significant amount of data as well. So there is a need for this repository function to be looked into, to be one of those factors that would catalyze the utility of wastewater surveillance in the continent. And so finally, I just want to end by acknowledging our funders and others who have contributed significantly to our lab. Would quickly acknowledge the Rockefeller Foundation, the Africa CDC, and BMZ, who are actively supporting our lab and research activities in general. And also, I would like to acknowledge the ILRI genomic team who help us to do this work. Thank you.
Thank you very much, Dr. Sam, for that very comprehensive introduction to wastewater and looking into the merits and demerits of it. I can see a lot of questions about the technicalities around it in the question and answer panel, but I think we'll take that after Muklid's presentation. So colleagues, we go now to our second presenter, Dr. Muklid Yusuf. So Dr. Muklid will delve into the technical aspects of wastewater surveillance, and he will share his experience from a South African perspective. Dr. Muklid, welcome, and over to you.
Thank you, Sarah, and thank you, Africa CDC, for giving us this opportunity to showcase our experiences in the wastewater-based epidemiology for SARS-CoV-2. Today, I'm giving this presentation on behalf of my colleagues at the Center for Vaccine Immunology and on behalf of the Sussex Network in South Africa. So the outline for this presentation are going to be 5 points. The first one is the beginning of the Sussex Network from polio, environmental surveillance to SARS-CoV-2. and then talking about the methods, how do we perform and report qualitative, quantitative, and genomic testing for wastewater, and then the results of SARS-CoV-2 quantification and sequencing, and then conclusion, how can we make interpretation of these test results, and finally, the future for the wastewater-based epidemiology in South Africa. So just to start by giving a bit of history of how did we all start that. So in June 2017, the NICD in South Africa has been starting doing poliovirus environmental surveillance. And that was based on the recommendation from the WHO, as the guidelines has been saying that environmental testing for poliovirus can be used as a complementary tool for the acute flaccid paralysis that looking for the poliovirus. And since NICD is a WHO accredited lab and the regional reference lab for the WHO, we started doing this since 2017. Soon after the start of the COVID-19 in April or May 2020, and globally, we found a lot of studies talking about the concept of the ES that could be used as a complementary system for the surveillance of COVID-19. So not long after that, in January 2021, our own South African Medical Research Council published the first proof of concept that SARS-CoV-2 can be detected in wastewater after lots of research studies have proven that it could be shed into the stools and therefore can be detected in wastewater. So in March, in our labs at the NICD, we start testing the wastewater treatment plant samples that we get from the polio program. So we started testing it for COVID-19, for SARS-CoV-2. And at the same time, the Water Research Commission has started its project in collaboration with the SASC network, including NICD, for monitoring SARS-CoV-2 in wastewater. That was the first phase that have a number of sites. And then in August, the second phase with increasing number of sites has also been added. In November 2021, we at the CVI received the first grant from the Gates Foundation, and that grant was specifically being targeted to do the sequencing and the molecular characterization of the virus in the wastewater. And recently, we received additional funding from the WRC and the GIZ in Germany to be able to expand the number of sites and the number of samples. So in total, this is currently what the status of all the sites in the country. So we have about 95 large wastewater treatment plants across the South African urban metros. 7 testing laboratories have been used into this, and our activities includes determination of the SARS-CoV-2 levels in wastewater and the genomic sequencing of the wastewater. How do we do this method? So as Dr. Sam indicated, we use 2 different methods for collection of samples. We use the passive sampling and we use the grab sampling. Then the next step will be that during the concentration of these viruses, we maintain the sample collected into the 4 degrees in the fridge, and then we use Centricon Plus filters for doing the concentration of the viruses, and this followed up by extraction of nucleic acids using the Quagene extraction kits. And finally, we do the detection viral genetic material using the RT-qPCR from Seegene. So all this will be then ended up into 2 results, which is the qualitative detection, whether the virus was present or not. And then we also do the quantification detection, which is we construct a standard curve, and that standard curve has been used with plasmid from the N gene of the SARS-CoV-2. And then we serially dilute that plasmid to be able to construct a standard curve whereby we can calculate the unknown samples for the amount of the genome copies per mil per each sample of the SARS-CoV-2. How do we display the results? I mean, after we do all this, then this graph is just showing similar graph to what Dr. Sam was showing as well. I'm just going to explain the details here. So in the x-axis, we have the epidemiological week, so it's a time. On the y-axis to the left, we have the clinical cases, the number of clinical confirmed cases. And on the right side, right side of the y-axis, we have the copies per min in logs. So this is not the absolute numbers, but copies per min in logs. In the different colors here, we have the different wastewater treatment plants. So this color is different from this color, indicating 2 different wastewater treatment plants. But the entire graph will be showing the results of a sub-district or a district in a certain geographical area. And then the other part of the work that we do is the genomic detection. So we identify the variants, the different variants of concerns that are circulating in the wastewater treatment plants. How do we do that? So there's a number of methods that we follow. The first one is by amplifying the whole genome of the SARS-CoV-2 using a modified Sinai protocol that was adopted from the Arctic protocol. So the Arctic protocol was the first protocol came to sequence the SARS-CoV-2 from the clinical samples. So we use some modification of that. And then we sequence Illumina using Illumina COVID-seq test. And then we do the bioinformatic analysis using the also Arctic protocol for bioinformatic tools. And we host that in the Galaxy project. And then just to explain, because of the nature of the of the sample of the wastewater, as Dr. Sam also explained, we don't generate consensus sequences from our FASTQ files that generated from the sequencer for reasons that we understand. This is not individual samples, but this is a population level samples. So it's not going to be meaningful to have a consensus sequences from samples that have been composed of different viral particles from many, many individuals. Therefore, we use a mutation analysis approach. How do we do that? We generate a list of mutations that we identify, and those mutations will be linked to, will be called unique or signature mutations. So each variant, whether it's alpha, beta, gamma, or omicron, will have a mutational profile. And we then look into those mutations and identify mutations that were uniquely found in specific and variant. And then we use our own codes that were developed in Stata and R to develop this master sheet that have the metadata plus having the sequence, the mutation that were detected. And eventually, how do we display these results? Similar like the quantification data, we have this graph that's showing the x-axis, the epidemiological weeks, and in the y-axis, we have the mutation frequency. And the mutation frequency will be showing the major and minor population. Because we're doing Illumina sequencing, so we generate these data. We generate that each mutation will be present, which frequency it would be, whether it's high major mutation or minor mutation. So in this graph, the blue here, or the color indicated the variant. So the blue is a beta, the green is a delta, and the purple is a C12 lineage. And then the shapes will be indicating the mutation. So because we picked up mutation, but we know each mutation is linked to each variant. So in this case, we say this is an evidence of the presence of beta in that time. And here it's an evidence of the presence of delta in that time. So then what do we do after we generate all this data? Then the partners labs from the SASIC network and the NICD, they all collect samples and collect data and generate results. And then all the partners send these results to the NICD on a weekly basis. And then our team manually enter the results and generate graphs. And then how do we disseminate these data? How do we communicate? We publish a weekly report on the NICD websites, talking about the wastewater epidemiology results. And then we also have our dashboard that was hosted in the NICD website. And also we have the social media platforms for most updates. Just showing a little bit of results in the surveillance. I'm not be able to show everything. I'm going to focus on the Gauteng province, and this is the city of Tshwane. So this graph, as I indicated earlier, showing the levels of SARS-CoV-2 in wastewater. So as you can see here, this high peak of the clinical cases was during the third wave of infection, and the second wave here was during the fourth wave of infection. So we can clearly see the numbers or the copies of genome copies per mil in the wastewater of SARS-CoV-2 were corresponding to the increase in the clinical case. So as you can see here, before the third wave, the clinical case was a little bit low, and as well as the copies per mil in the wastewater. But we see here the increase of the copies per mil in the wastewater has corresponded to the increase of the clinical case. Similar thing happened during the interwave between third and fourth wave infection, where the decrease happened in the wastewater treatment plant was corresponded to the decrease happened during the clinical isolates. And similar again, the increase of wastewater copies per mil has happened during the in the same time of the increase in the clinical cases. So that's just to show the indication of that, we can say that there is correspondence between the clinical cases prevalence plus with the copies per mil of SARS-CoV-2 in the wastewater treatment plants. If we look at the variant analysis, as I explained earlier for this graph, we can see here this color. This color is indication of presence of beta in a different ratios. So this from 60% up to 10%, but it's indication of the presence of the beta, of the delta. And if we look at these times, epiweek 2021 to epiweek 31 in 2021 last year, those at the time when South Africa was experienced with the third wave of infection that was predominant by the Delta variant. So that's also indication of the corresponding findings. And in addition to that, to confirm these results, in the fourth wave of infection, we found that from epiweek 44, Omicron has been dominating in our wastewater. So we don't find any more Delta here, but we only find Omicron. And just to say also during the fourth wave of infection, until now, Omicron is a predominant variant that's circulating in the country. And we even found the subvariant of Omicron of BA4 and BA5. We found similar results in the other cities. So if we look at the city of Johannesburg, we don't find any differences between these results and the previous results of the increasing numbers of copies per mil during the third wave and the fourth wave, and the corresponding between the wastewater treatment plants levels with the clinical isolates as same as present of the Omicron mutations during the fourth wave and after also the fourth wave of infection. City of Ekurhuleni have the same results, showing that increase of the wastewater treatment plant SARS-CoV-2 levels during the third wave, fourth wave, and decrease in between, and the presence of Omicron. So this is how we do generate our results for the entire data set, for the entire wastewater treatment plants, but for the sake of time, I'm just showing a few examples. The second part of analysis that we have done is using a Freyja tool. And here we're trying to show the overall proportion of the variants of concern and lineages that we detected in the wastewater treatment plants in South Africa. So this work has been done in collaboration with Scripps Institute from Christian Anderson and Josh Levy. They have developed a tool called Freyja, and the Freyja tool has the ability to determine the proportion of each variant in specific in one sample of wastewater treatment plant. So in this graph, you can see the color here. For example, this yellow here is indication of the variant B1351, which is a beta variant. And that was during the time of when the beta variant was circulating in the country. And as you can see in this, the number has been decreasing this time, which is probably during May and June, July time, when we can see the Delta start to appear. And this color, these two darker and light green color are indication of the Delta. And that was followed by the presence of Omicron, which is in the blue and the orange. And so it's actually also confirming that the methodology that we have used and has been able to confirm the same results that has been detected in the clinical samples. This graph only ended in January 2022, so I wouldn't be able to show the BA4 and 5 that's currently circulating as subvariant of Omicron. But we took this tool and we use it further for the monthly basis, not the daily basis. So here we can see that these variants of concern how are they distributed on the monthly basis in the country. And we can also confirm that the presence of beta in April, and then the decreasing in numbers in May and June. And then we can also see the pickup of the Delta starting from June, and then the transition again from Delta to Omicron. There was no data in September, due also to the low spike low coverage in terms of the sequencing coverage that we produce. And very interestingly, also we have this graph that's showing even into the lineage level. So it's easy to see the variant of concern in one graph because there are low number of variants of concerns, but the lineages, there are just so many lineages of SARS-CoV-2 currently that has been found or detected. But the tool was able also to be able to demix all that happening in the wastewater sample and give us an indication of proportion of each of these lineages. So the interesting future work that we're trying to do here is to try to confirm that all these lineages that we found in our wastewater treatment plants were indeed found in clinical cases. And if not, then this is a question that we will have to answer. Third part of the analysis is working on the pattern of the mutations that we found in the spike protein. So we generated this heat map. So basically the heat map is composed of the Y axis is time. So here is the second wave, here the third wave, here the fourth wave. And here is the spike protein, the entire spike protein. So we're showing only the mutation that we picked up in the spike protein. And the darker color, the red color, is indication high frequency of mutation, the yellow indication low frequency of mutation, and the no color is indication of no mutation at all. So what we can see, we can identify a certain specific mutational pattern for each variant, and therefore it's for each, it could be also for each wave. So you can see there's a pattern of mutation here and there and there and there. But what happened from this time to the next time, which is from the third wave to the interwave, that we lost all this mutation. And we think this is also could be attributed to the fact that there was low coverage of spike has been sequenced. And the reason for that also could be because of the low number of cases and therefore low number of people shedding the virus into the wastewater. But when we compare the third wave, this mutational pattern to this part, which is the fourth wave, to this mutational pattern, we can see the difference. If we look at the NTD region here, we see there's this pattern of mutation that has been lost here. And in the RBD region and the fusion peptide region, there's also a number of mutations that were picked up. So, I mean, obviously this could be explained because here it was Delta and this is Omicron. So, and Omicron has more mutations. So that's why we can see these differences. But what we wanted to say in potentially, could be a method to be able to pick up new mutations that there was not enough time yet to accumulate in clinical cases and show as new lineages. So the wastewater will be able to, if there is a new pattern happening in the future, then we'll be able to use as a tool for a warning system to say a new mutation or a new virus is coming up. So then the fourth part of the analysis, we took the same mutation that we described in the heat map and we applied a 1% cutoff. So we did not use all the mutations. We just used all the mutations that were present at more than 1%. And we tried to look into the details of these mutations. What actually are these mutations? And then we divided them to two parts. The one, we call them the common mutation that we found mostly in the clinical isolates in the country or globally. So they were not surprised by seeing these mutations. And we tried to establish the prevalence of this mutation in wastewater versus the prevalence of this mutation in clinical isolates. And we established this graph. And there is no surprising results here. So we found that most of the mutations that present in the clinical were actually higher than the one that present in the wastewater as expected. And finally, what we also did here is look at the mutations that were also detected at more than 1%, and they were uncommon. So those are surprise mutations that we couldn't find them regularly at the clinical cases in GZ. So the question is then, what are these mutations? So firstly, we established the time when these mutations were started. The x-axis is showing the time and the y-axis is showing the different mutations. So we're showing when it was started and how many times those mutations were picked up. So then the next question would be, are these mutations real? And if yes, what are the phenotypic effects of this mutation if they were present in humans? So in conclusion, correlation between SARS-CoV-2 levels in wastewater and case burden in waves, all the 4 waves, they varies between test methodology, and it's also better to have more sensitive methodologies. And we also say that there is no predictive rule yet to be able to identify or predict a new wave, but it's something that we're still working on. And for sequencing, we say that it's corresponded very well to the clinical and variant data, and it's able to provide comprehensive details on the lineages found in population. What is in the future? What is the next steps? What are we planning to do is to reconfigure testing network. We're planning to adopt the standard sensitive methodology and to increase the testing frequency in order to be able to support the early warning system. And we're also planning to collect larger variety of metadata that will also help us to do the normalization, like the flow rate and the population size. And we also wanted to understand better the rate of the degradation of the virus due to the dilution factors like the environmental factors like the rainfall, for example, which is something that yet we haven't done yet. And for the sequencing, we also wanted to understand the correlation between the wastewater genomic findings and that relative to defined clinical cohorts. And the second part of the future, what we wanted to do is test it for other pathogens. We have plans to test measles, polio, and other pathogens in wastewater. And I will stop here and thanks all the core team and the Center for Vaccine Immunology and our partners and funders. Thank you.
Thank you. Thank you very much, Dr. Muklid. Colleagues, we've come to the end of the two presentations, quite very interesting questions in the Q&A panel, and I will just start asking by one from our side. Maybe perhaps Sam or Muklid, whoever wants to take this question. So in the wake of the emerging and reimagined pandemics, such as the monkeypox that was recently declared a pandemic of global concern, and perhaps Marburg case in Ghana, and more recently, the Ebola case in DRC. Could you share your reflection on how the research you've been conducting on wastewater can prepare us for such scenarios in the future?
Okay, thanks, Sarah, for that. Well, I think there is a possibility that wastewater surveillance can still be applicable in that case, but there's still need for some work to actually confirm that we can detect, say, monkeypox virus in wastewater. I know that in the wake of developing wastewater surveillance, there were doubts that several pathogens could not be detected in wastewater, particularly enveloped viruses where people think are very delicate, etc. But actual data shows that we are able to detect this. Our case in point here is SARS-CoV-2, which is an envelope virus that people thought we were not able to detect, but is detectable. So I think it begins by preliminary studies that can show that this can be detected. And if that is the case, then it would definitely be useful in helping such cases. But it also has to depend on the type of pathogen where it infects, etc. Because not every pathogen can be detected in this system. So I think it's just preliminary work to show that it is possible, and then when it is possible, then the tools are ready to be deployed. Notwithstanding the limitations that I've mentioned in my talk.
Thank you, Dr. Sam. So, so the next question, this one I think is for you, Dr. Muklid. Sarah Downs from South Africa is asking, the South African wastewater surveillance team, I think it's at the MRC, have done a project that involved non-seaward wastewater, which I think is very interesting in the African context. So where communities, where sanitation networks are not in place, you're talking about small rivers. So this was done for detection of SARS-CoV-2. But would this work for AMR panels or sequencing, or would you anticipate too much dilution or interference?
Yeah, so basically we also did this recently. I haven't shown the results in this presentation, but we recently also collected samples. In collaboration with Water Lab, we collected samples from rivers in Johannesburg. And the first thing that we noticed is the low number of positive cases comparing to the positive cases from wastewater. So two reasons could be also because of the current status of the pandemic in the country, the epidemic in the country, that it's low numbers. And the other thing, as we indicated, the dilution factors, because this is a running water, this is a river water, so it won't be concentrated as in the wastewater. So yes, it is a very good example of utilizing different samples because of the context in the continent. And I'm sure AMR or any other tests that we would like to do in all other pathogens is also possible to do, but we must be aware of this limitation of the dilution that we might or should expect.
Okay, thank you very much. So this question, the next question goes to Dr. Oyola from Queen, and he's asking, I'm surprised that you're able to detect SARS-CoV-2 in wastewater. SARS-CoV-2 is an RNA virus, and the RNA molecule is very unstable. Are you detecting viral RNA or DNA after it was inserted into the human genome hosts during infection?
Okay. Yes. So here we are detecting the RNA from the SARS-CoV-2 virus that is in the wastewater. So SARS-CoV-2 virus can be excreted either through urine or feces, and when it gets into the wastewater, we are able to actually detect the RNA. So when we concentrate the wastewater during processing, we actually extract RNA, and then we convert that to cDNA and to be able to carry out the detection. So we actually detect RNA, not DNA, for SARS-CoV-2. We are detecting RNA. Thank you.
Thank you. Thank you. And then we have this question from James. I think this one either of you can take. What are your thoughts on the utility and scale of using wastewater surveillance for determining transmission networks?
For me?
Yeah, you can go ahead.
Okay. Yeah, so I think what I was trying to show in the data there is actually the actual utility. Because when you pick wastewater and you've done your sampling correctly, you are covering a particular epidemiological zone, a population, and with actual quantification of the amount of virus that you get over a period of time, you are able to actually figure out what the transmission rate is of the network within the population. But when you want to look at the transmission dynamics, etc., then you will be required to pick several samples from different populations, and then you do, after detecting it from the wastewater, you then do a thorough characterization, maybe sequencing of those samples, and then you are able to relate from the sequence information, you will be able to relate the transmission dynamics. So for example, this virus that we detected in population A, how is it related to the one we are now detecting in population B, based on analysis of the mutations that you see there. So you are able to detect the parent virus and then the other one. Yeah, so with that, you are able to look at the transmission within populations. Yeah.
Okay. Thank you. Thank you very much, Sam. So I think in the interest of time, I'll just take two questions. And then I think the rest we will have to get in touch with the presenters personally. The next question is from Richard, and he's asking Muklid, from the Freyja tool graphs, you showed a recombinant Omicron lineages as a category. Maybe a stupid question, but if you're not generating consensus genomes, how can you identify recombinant lineages?
Yeah, that's a nice thing about the tool. The Freyja tool generates algorithms. So the tool itself generates algorithms. It defines the mutations per each variant or lineage. Each lineage or variant that has been deposited into GEZIT have specific mutations or mutational profile. So what the tool does, it takes all these mutations for the whole genome and then generates a matrix for each lineage. So each lineage will then have a defined matrix. Then the tool will take the samples from the wastewater of the whole genome sequencing and do the mapping and everything. And then it looks onto these matrices. And then with the matrix itself, it will be able to identify whether that was that read specifically. So it takes each read and look at the matrix, and then each read will be assigned to certain variant or lineage. And then in total, what it does, it then it does do the accumulation of all these reads to have a proportion of each variant or lineages in sample. Thanks.
Okay, so thank you very much. So I think I will take the last question, which is from Geoffrey Kumwenda. So he's asking, how long do SARS-CoV-2 RNA last before it degrades in an environment like wastewater? And he's asking this just to learn, wouldn't the detected N gene be an accumulative representation of current viral shading and also previous genes or previously partially degraded genes that are still circulating in the wastewater system? So it's really about sensitivity. I think, Sam, you can take this question.
Yeah, so it is possible, but the nature of the wastewater, when we are talking of a flowing wastewater that is actually coming from a particular population, when you pick the samples within a specific times, there is a way in which you are able to know that this is a sample that represents a particular time. Okay, because it is not, we're not talking of a wastewater treatment plant where we have accumulated sewage water. We are talking of an actively running wastewater. So you are picking at a manhole of what is continuously running. So there is no possibility of say that this is a virus that has been accumulating in this point. You see what I mean? So that is how we look at it. So every time you pick a sample, you know that it represents what has been excreted or deposited in the sewer line at a given time. That's why it is spatiotemporal, space and time sensitive. Yeah.
Okay. Thank you very much to you, Dr. Sam and Dr. Muklid. I see we've got very interesting questions in the Q&A panel, and perhaps we can take this on your emails.
So, sorry, Sarah, let me just maybe quickly, there was a question in the beginning which I did not manage to answer, the very, very fast, to talk about the ethical issues. The ethical aspects that I mentioned there is that there is need to train the public health on the actual meaning of the data that comes from the wastewater. So this is just to protect the reporting so that the media does not take it in a different way that can cause ethical issues. There is a way in which the wastewater is interpreted because it is a population, it's not an individual. So I think that is what I was referring to in terms of ensuring that the ethical issues are taken into consideration.
Okay.
Thank you.
Thank you. Thank you very much, Dr. Sam. So there's a guy called Abdullahi who's commenting about sharing the materials. Just to notify you that we do actually share a link of the recording to all registered participants. So you'll be able to get a recorded version of this presentation. So I think, ladies and gentlemen, this marks the end of our webinar today. Dr. Sam and Dr. Muklid, on behalf of everyone here today, thank you very much for taking time to speak to us on this very interesting and important topic of wastewater surveillance. On behalf of the Africa CDC, I take this opportunity to thank you very much for making time to attend this webinar. Please be advised, it's a bi-weekly activity, and we are going to have the next one on the 7th of September, 2022, where Dr. Ife Anyebo will be talking about malaria molecular surveillance in Nigeria. Stay safe and do have a lovely rest of the day. Thank you very much.
Thank you, Sarah.
Thank you. Thank you.
Bye.
Bye.
Recording stopped.